Hello, everyone. Welcome. My name is John McLaughlin. I'm a research scientist at Rutgers University. My job involves plant pathology and genomics, and I study the blueprints of how plants defend themselves against disease. And the reason I'm here today is trying to figure out how to get supercomputing into more hands at the university, regardless of where they live, their family's income, or what laptop they can afford. This talk is called Local Edge Computing in the Online STEM Classroom. And the subtitle gives you the punchline. So, opening the door to supercomputing for students. Now, of course, we're awash in computing with cloud computing. There's avenues on Apple and Windows systems to running Linux. For example, Windows, you can run the Windows subsystem for Linux and install Linux separately and run it from there. But today we're really thinking about in a classroom setting and how getting everybody on the same page. And one thing that has come available is sort of like a Raspberry Pi type unit in size, but housing a high performance GPU, which is the NVIDIA Jetson Nano Super. And that's what we're talking about in context to doing bioinformatics, but you could apply this to many different other types of sciences and other fields. So the invisible wall. Every time you design an online STEM lab, you hit the same wall. You don't know what computer your student is sitting in front of, and you write your lab for your particular machine generally, and then they try to run it on theirs. And sometimes that breaks. The first 20 minutes of office hours often become tech support instead of science. And so that's where there's a hardware gap in some cases, and that's been there since online STEM education has existed. What if you could mail or direct the lab bench to the student? So in this particular case, we're imagining a pre-configured around $250 device that gives every student, whether they own a $200 Chromebook or a $4,000 gaming rig, or access to online cloud or supercomputer access, for example, here at the university, the same computational power, the same software, the same research environment. It standardizes it. No installation, no server waiting periods, no internet dependency. And that's what this talk is all about today. So my journal, I defined that, I entitled this From Turtles to Tensors. So when I grew up in the 80s, we actually had an Apple store in our town, and they had the Apple II, and my parents enrolled me in a class there, and we learned about Logo. I'm not sure how many people in the audience remember Logo, but that's an early Apple programming language that you program to move in certain patterns, and you can make artwork or just as a way of learning computer programming in a very basic sense. And then going from that, which really introduced me to computer programming at the basic level, going to grad school where we, for example, hit a bottleneck, or when we're doing gene mapping, we hit something called the permutation wall. This statistical analysis at the time would take over 24 hours, and the results were obviously limited by computational power. This was resolved later with new generations of Intel CPUs, but it made me appreciate the power of compute that you have to solve scientific problems. So the session objectives today, really to identify how edge computing reduces internet dependency and server costs for remote learners. Discuss how using low-cost hardware can improve digital equity in STEM courses. And we'll talk about some designs for hands-on, offline capable technical modules for bioinformatics and AI curricula. So a hardware gap exists in online STEM. So some cases we're grading hardware adjusted achievement, not raw skill or understanding. And a lot of work can be done to reduce this. So sometimes there's an institutional bias where the internet speed is an unspoken prerequisite. Sometimes there's a broadband poverty where remote students are capped by their connection, and thankfully that's being reduced. And institutional clusters can serve as scheduling bottlenecks. So this edge computing example using a Raspberry Pi or this Jetson Nano mini supercomputer, this is a case where we can think about standardizing their environment. Every student gets access to identical setup or uses a supercomputer directly. There's basically a zero install that it can become preloaded tools on day one, and offline power, no cloud dependency, no server queues. So let's learn more about the Jetson Aura Nano Super. Again, I just think this is sort of like the Raspberry Pi of supercomputer world. Fairly low price at under $250. AI performance 67 tops. This is enough to run AI models locally offline. There's 100,024 CUDA-core GPU performance units. This allows for parallel processing of data analysis. A relatively modest amount of memory at 8 gigabytes, but this is enough to run bioinformatic tools. We're not gaming with this instrument, this computer. And other benefit is that it runs Ubuntu. Ubuntu is the same Linux as a lot of research servers use, and it provides a great learning environment for Linux. And the power you can run from all the way from 7 to 24 watts, and the 25 watts rather, and the 25 watts allows you to do something called like super power mode. So we're not asking the students to run a server room. This device will become pre-configured, or they could just call into one that exists at the professor's desk or in the lab. And the other thing that this uses the same core NVIDIA universe as the Ampere GPU family as the data center systems, though it's just scaled for a desk. And so why does this work? What's the theoretical framework? So if we can think of three things. We can think of TPAC, which is the technological, pedagogical content and knowledge. The Jetson integrates technology knowledge, Linux, GPU computing with content knowledge, genomics, statistics, other bioinformatics tools through pedagogical design, scaffold modules, formative assessments. And then we can think about universal design for learning. Standardized hardware provides multiple means of engagement, tactile, visual, analytical, representation, where we're looking at code, visualization, even sensor data, and action and expression, Jupyter, Git, Canvas. And we can think about productive failure, where the students have desirable difficulties. The 8 gigabyte memory, for example, is a constraint, and normally you'd think that as a limitation, but for teaching, it could be considered a benefit. It's a curriculum design tool for that limitation. Students who learn to optimize for constrained hardware develop deeper computational thinking than those who have just unlimited RAM, for example. And so just some economic shift, dry labs versus wet labs. Now, of course, wet labs are essential to university education, but today we're just thinking in terms of taking some of that wet lab information and performing dry lab sessions with it. So, of course, wet labs can be expensive with the PCR reagents and things. The biohazard and chemical waste disposal requires, of course, physical presence and vanishing funds after the labs are finished sometimes. The dry lab, where we're talking about bioinformatics today, relatively low cost, $249 for the device, or it could be free depending on if grant funded or the student just calls into the device by SSH. We can reuse the hardware. We estimate that there's at least a 4-year lifespan for these. And they also offer remote, field, and online flexibility, and also the possibility of building funds for permanent infrastructure. So I'd like to present a case study. This is something that we did in our lab, where we were looking for novel proteins in public databases. And I've entitled this Finding a Needle in 12,880 Haystacks, for example. So this is something where we can have a scientific discovery that we made in the lab and present this to the students for them to recreate that finding. So, for example, in the lab, we study these non-specific lipid transfer proteins. These are basically plant defense molecules that are ubiquitous in plants. And we can think of them as part of the plant's immune system. And our lab discovered never before reported variant by screening about 13,000 of these proteins computationally. And in this particular exercise, we can have students reproduce the exact discovery on a Jetson using methods that they program and they access public databases directly. So, for example, what we searched approximately 13,000 proteins in this non-specific lipid transfer protein superfamily, and we found approximately close to 12,000 standard one domain binding, the lipid variants, and that basically established a baseline. And then we found 35 rare multi-domain variants that haven't been reported on. Some of them have been known, but definitely understudied. And we also found one unprecedented, not reported, four-domain variant. And I have a picture of that labeled H on the top of the slide. This is a protein that has four domains that are able to bind lipids potentially independently. And so that has a potential novel function, and that's something that we'd like to learn more about. So the academic return on investments is students master the same pipeline that we used in our Rutgers lab using Python scripting, database searching, and protein structure prediction on their mini supercomputer. So what else can students do? Let's think about 3 live demonstrations. So let's think about demo 1, where we have DNA base calling. We can do DNA base calling as it occurs on the instrument with something like the Nano. This is where the student converts raw electrical signals from the DNA sequencer into readable genetic code. That allows the student to see how things are done in real time, and it can solve a CPU bottleneck and perform this task from 10 to 50 times plus faster compared to traditional CPU base calling. Protein folding. There's a program, ESM Fold, that can be used in Linux. This predicts the 3D shape of a protein from its amino acid sequence. And this helps students interpret that 2D code into 3D structures. And we can also put confidence-colored 3D structure information using programs available from the protein database group here at Rutgers, and using like a heat map of a certainty. The students can compare the speed of this using GPU versus non-GPU methods. And then a third live demonstration could be population clustering, for example, k-means. This is a case where perhaps you'd group 300 individual data points into subpopulations based on genetic markers, for example, from a genetic mapping study. And here is the idea that iteration is the core of some science, where students watch centroids converge in real time using the GPU compute. And this is just, for example, using the programs that pre-exist, and you can compare using GPU versus non-GPU methods. And so basically each demo is are you ready to teach this skill moment? This is all pre-existing technology, of course, but it does introduce the students to the NVIDIA universe of programs and that wider universe of Linux bioinformatics. Constraints as pedagogy. So why is 8 gigabytes a better teacher than 1 terabyte? This goes back to the productive failure method of applied to computing. So the infinite trap server. So sometimes over provisioning hides inefficient code, and just adding more RAM replaces understanding. Students become more cloud runners, not architects. And sometimes if you have a crash of, for example, 112 threads going, that can be a high stakes, slow learning situation. Whereas in edge computing, using something like the Jetson, it forces mastery of memory management and profiling, requires quantization and optimization of knowledge. It allows the students to have root access to the Linux system. So if they break it, no big deal. You can just reflash it in a small amount of time. And so this is what I would say low stakes, high learning. So you have the freedom to make mistakes and to fail. So let's think about a module design for week 4 where we're looking at protein structure using this type of edge computing. So let's think about components. A learning objective where the students will predict and evaluate protein 3D structure using the ESM Fold on their Jetson device. The pre-work, we can think about what is protein folding, the computation involved, the history of that, and why does shape matter? What does that mean biologically? Hands-on activity, of course, running the ESM Fold on the proteins that they identify from their database searches. We can color those folds by confidence using the tools from the protein database. And we can compare that to AlphaFold database entry computations. Reflection. What does the confidence coloring tell you about which predictions of the proteins are which regions of the proteins are well predicted? And we can have a peer response discussion about that. And then for example, a rubric you mentioned, a technical execution 40%, for example, biological interpretation 30%, and a written reflection 30%. So it really involves the students from A to Z. So what is the student experience using something like this, using a Raspberry Pi or a Jetson? So basically going from unboxing to Canvas. So what it would look like in practice. On day one, we would unbox and boot the system. We would open our laptop browser, for example, and Jetson SSH to the Jetson. That's also a possibility. Week two, we could perform these k-mean analysis, do the bioinformatics. And then in week four, we could fold those proteins that we identify with bioinformatics using ESM fold locally. We could visualize that 3D structure in Linux with the Linux system and push that information to Git and then Canvas. And then one student that I worked with, his voice, I'll just add, he said that when I first got this thing, I thought it was going to be a toy. It's smaller than my textbook. But the first time I watched ESMM fold generated protein structure, I realized I just did something that would have required a server account and a queue several years ago. That's the kind of doing it on their own system. That's the kind of experience I can build upon. So what we're talking about is shifting the capacity of the scientist. So the paradigm shift, data gravity. So the old way, maybe upload raw terabytes to the server. The Jetson way is potentially process locally, upload results within minutes or in real time. Agile discovery. So post-exhibition analysis perhaps takes weeks. Here, you could perhaps run this in a real-time OODA loop, which is observe, orient, decide, and act. Whereas if you want to make changes on the fly, the students can do that. Resource awareness, knowing what your resources are and what you can and cannot do. So for example, if you know you're limited in RAM, how do you design your systems to handle that? And then also realize, we might need more RAM to realistically do these analyses in the future. In sensor fusion, DNA data is separate from the environment, and using local edge computing, you can have, for example, DNA sequencing, taking temperature and GPS data all in one device. And then this idea of a digital twin works on my machine. So basically, identical hardware, the Jetson way would be identical hardware for every student, so everybody's on the same page. This is just makes the class much more unified in that particular aspect, especially when it's data intensive and computationally intensive. So let's give an example of fusing DNA with environmental context. This idea of Internet of Living Things, a novel research platform. So imagine a case, for example, my wife does real-time stream data collection where they measure temperature, water speed, and a number of different things like that. So you could imagine a case where you combine that with DNA analysis for a particular problem. Here you can have these different low-cost sensors, although the sequencer is at high cost, but relatively low-cost sensors hooked up to your unit and collect that data in real time while you're doing other computations like sequencing DNA or RNA. So the idea here is every genomic read becomes a multimodal sensor event. You'd have DNA sequence plus temperature plus humidity plus GPS coordinates. So you can imagine a case where you have an environmental event which changes over time in terms of the microbes you're sequencing, for example. And this would provide a publication opportunity for the students as well, for the group or individual student. This also introduces the students to this three-tier model idea. So going from edge to cloud. So the tier one would be this local edge, this Jetson Nano, which is lab owned or student owned, and allows them to do base calling, protein folding, motif scanning in k-means, and other bioinformatic analysis right on their desk. And that can lead to better understanding how institutional servers work, where you have Red Hat, multi-threads, over 100 threads going with a great deal more of RAM to deal with. That would be the case where you're doing genome assembly, GWAS, which is a type of genetic mapping on a population level, and population genetics. And then leading to research clouds where you have even stacks and stacks of GPUs working together. This is the case, for example, when they are doing AlphaFold protein modeling on databases of protein information. And I think this type of thing, being exposed to this, helps students understand these different levels of high parallelism computing at different GPU and RAM constraints. The other value is this privacy first AI. So edge is a practical and ethical choice for some data sets. So if we think about the cloud model, the student data goes to a third-party server in some cases. There could be some compliance risks where that if you're working with a company, for example, you're not permitted to do that. And the institution or the student is dependent on vendor uptime. Whereas the edge model, all that data stays on the student's device. Local large language model tutoring, for example, is possible. And it's compliant by design. No data will leave that student's computer. And also the student owns their own learning artifacts. So edge computing can be seen as not just a performance decision, but also a privacy architecture. Data sovereignty is a learning outcome, not just a compliance checkbox, which can be another important learning factor in this, what we're presenting today. So what's the assessment like rating the sovereign learner? So we can evaluate both the answer and the computational literacy to get that answer by looking at the local compute, their Jupyter notebook, their Git push, and finally their Canvas grade. So the formative process, so that Git commit history shows problem-solving process. Did they debug their own Docker container? How did they solve problems? The metric would be self-efficacy, troubleshooting, and iteration. And the lesson could be designed to introduce problems on purpose to see if the student can solve that. And the summative output, the Jupyter notebook with methods and results can be one output that can be graded. The statistical biological accuracy of the output can be measured. And the metric would really be the content mastery and the computational skill required. And how does this compare, like the Jetson versus the alternatives? I mentioned before, in many cases, if the student's computer already has a high-powered graphics card, they could just use a Windows subsystem Linux install to do this. And just use the same version of Linux and we're off and going. But the benefit of having a class use the same system is it gets them used to using something like an edge computer like the Jetson. And I won't go through all of these things in the table, but basically it compares the Jetson to the Galaxy to the cloud and then back to the wet lab. So if we talk about accessibility and inclusive design, we can how do we ensure that every student can engage with a physical computing device? So if a student has motor impairments, the way we're thinking about this particular case, all interactions are via an SSH terminal or Jupyter browser interface. There's no soldering, no fine motor assembly required. And we're also able to use general purpose input-output sensors. Those are optional for enrichment, not necessarily required. If students have visual impairments, these terminal-based workflows can be screen reader compatible, can be made screen reader compatible. Jupyter notebooks support alt text for figures, and high contrast terminal themes are pre-installed. Cognitive load, the pre-configured SD card eliminates installation complexity. The Docker contains reduced setup to single command. And the scaffold modules build from simple to complex. So there's a range of complexity that can be built in. Economic access. Course material fee model, where the student purchases this, is comparable to existing lab fees. Financial aid could be applicable, and a loaner pool for students with demonstrated need, or just simply ask the students to SSH into available units in the class is also a possibility. Rural and remote. This is offline capable by design. Students in low bandwidth areas are not at all disadvantaged. All compute runs locally, and then only that Canvas sync requires connectivity. So if there's limited connectivity, results can still be uploaded. And of course, if we're using a large data set, that also could be mailed on a large capacity drive, flash drive. So what are some honest limitations? What the Jetson cannot do? Some real constraints. So one constraint is that because of the low RAM, you cannot train large AI models. That training would be reserved for something like the NVIDIA Spark or something with more RAM. Can't run the full AlphaFold 2. That requires more than double GPU, gigabytes of GPU RAM. But we can run smaller models using this ESM fold and then apply AlphaFold servers for larger jobs. It does require basic Linux comfort with commands, but that's one of the benefits of this class. So the class provides Linux experience and command line interface. That gets students to the command line interface, not necessarily a GUI. And that can be built into like a 2-week Linux bootcamp in the syllabus. Fleet management, to preload all of those information to the SD cards, although that can be made fairly straightforward. And we're still generating student outcome. The pilot phase has not been completed, and we look to work with more students on this. And again, it should be very straightforward, be very clear rather, that this is not a universal replacement for cloud computing or high performance computing. It's just exposing the students to this tier 1 device where they can get root access, they can serve as a system administrator if they wouldn't want to and make changes. They can work to optimize software, work to optimize the hardware, and then if they decide to buy the unit, use it for other things as well, and experience that computational ownership. So how do we measure the impact of what a class like this would entail? So we could use a Likert scale. We could present this 1 to 5 scale to the students, and we'll just go through the survey items here. So, can the students, after they complete a course like this to measure computational literacy, can they explain the difference between CPU and GPU computing? They've done it firsthand, so it should be straightforward. Self-efficacy. Are they confident that they could set up a bioinformatics analysis pipeline independently? They will have in the class. Resource awareness. Do the students optimize code for memory constraint environments? So modules would be built where the code does not work as is, and they would have to optimize that code to work within 8 gigabytes of RAM. And self-directed learning. Can students troubleshoot a Linux system error without instructor help? And sovereign learning. Does the student feel ownership over their computational research environment, which I think is an important thing. And computational strategy. Can the student describe when to use edge computing versus cloud computing for a given task? I think that's important in our current environment. And digital equity. I believe my hardware setup does not limit my ability to complete STEM homework and coursework. So how might we finance this idea, this sustainability model? So a pilot phase could look something like this, writing to NSF or the NVIDIA academic unit. And we could start small with 5 to 20 student lab-in-a-box kits, and we would measure this self-efficacy and retention. The budget would be on the order of $2,000 to up to $30,000, depending on the sensor and the other peripherals that the lab wants to use. And that would build up to like a program phase, like for example, three plus years, where there could be an institutional fee for the mini supercomputer, or just have a buildup of them to hand out to the students to use. Ownership, the student could keep the device, or if it's self-sustaining, the student returns the device. We wipe it clean and start fresh for the next class. So our next step, as Dr. G.S. Hai from Indiana Institute of Technology noted in last year's RU Online Con, the gap between A-level and C-level work will only widen because AI literacy comes with its own privilege. This talk offers an infrastructure level response to that challenge. So I would hope if there's feedback to what I presented today, I hope you can get back to me either by email, or I've put up a Padlet sheet, a Padlet source that you can touch base with me on, and also on GitHub. You can see what we're doing there on GitHub as well. And I hope you'll connect with me. My email is there. And if any of this looks like it might fit some of your classes or just part of your classes, I'd be happy to talk with you more. And so the future of STEM is local. So I'm joking a little bit, but at the same time, I think something like a Raspberry Pi supercomputer like this Jetson provides can provide equity in infrastructure that we can build for our students, and then helps them lead them into graduate education or in bioinformatics or in biotechnology in industry. Well, they'll be exposed to even more compute power, but they'll at least have a base for the compute power that they learned using local edge computing. And with that, I appreciate your attention and thank you very much. Appreciate that.